Cancer is a major cause of morbidity and mortality in dogs. Approximately 25% of dogs will be diagnosed with cancer at some point in their lives. Lymphoma is one of the most common cancers in dogs. The monoclonal antibody is a biological therapeutic intended to aid in the treatment of lymphoma in dogs. This particular monoclonal antibody therapy was engineered using the Aratana therapeutics technology platform. These monoclonal antibodies have been engineered to recognize a specific target protein or antigen that is present on the surface of both a normal and a targeted cell. These particular monoclonal antibodies are canonized. In other words, they are uniquely designed to be compatible with a dog's immune system. The monoclonal antibody binds to the antigen on the target cell to ultimately help cause its destruction. The monoclonal antibody enables the immune system to identify and deplete malignant cells in several ways. The first mechanism of cell death may be achieved by complement-dependent cytotoxicity, or CDC. CDC is achieved when a group of plasma proteins, in combination with antibodies, causes destruction of invading pathogens and malignant cells. The complement system is activated by antibody-antigen complexes. The product of each reaction in the process starts the next event. As the monoclonal antibodies bind to their target receptors on the malignant cell surface, the complement protein C1 recognizes and binds to the tail of the monoclonal antibody, starting a series or cascade of sequential reactions. This cascading sequence of proteins ultimately creates the formation of protein-lined pores on the surface of the cell. This process is known as the membrane attack complex. These protein-lined pores penetrate the cell membrane, allowing the cellular content to escape, resulting in death of the cell. Monoclonal antibodies may also destroy malignant cells through a process called antibody-dependent cell-mediated cytotoxicity, or ADCC. This method of destruction involves other components of the immune system, including natural killer cells. When the monoclonal antibody binds to the target antigen on the surface of the malignant cell, the antibody-antigen complex attracts natural killer cells that detect the complex through Fc receptors. The Fc receptors on the natural killer cells recognize and bind to the species-specific canonized tail of the monoclonal antibody. Natural killer cells also carry granules filled with cytotoxic molecules. The granules are released when the monoclonal antibody is detected by the natural killer cell. Some of the granules penetrate and form pores that allow cellular content to escape. Others penetrate and destroy the cell's nucleus, ultimately killing the cell. Monoclonal antibodies provide a targeted immunotherapy that specifically recognizes the cell surface antigen expressed on the malignant lymphoma cells in the dog. Upon binding to the cell surface antigen of the cell, the dog's immune system is better able to identify and eliminate the malignant cells. Aratana Therapeutics has received a conditional license from the USDA for canine lymphoma monoclonal antibody as an aid for the treatment of T-cell lymphoma in dogs. Activity toward full licensure is currently being pursued. Monoclonal antibody safety has been demonstrated by administration of the recommended dose to dogs with T-cell lymphoma. There are no known contraindications for the use of monoclonal antibodies in dogs with T-cell lymphoma. In instances of hypersensitivity types of reactions, therapeutic administration should be suspended. Treatment may include antihistamines and/or anti-inflammatories.